Today we will be analyzing an mRNA sequence that codes for the Pokemon Charmeleon. Here is Charmeleon's DNA sequence that we will transcribe into mRNA. I read the top DNA sequence backwards to transcribe an mRNA that will result in a 5-amino acid protein. This mRNA sequence is the wild type and will result in a normal Charmeleon. The mRNA translates into the following amino acids: methionine, valine, arginine, phenylalanine, and lysine. With transcription comes room for error. For example, a single point mutation can happen. A single point mutation is one of 4 types of mutations and is defined as a substitution of one base for another. If a purine base is substituted for a pyrimidine base or a pyrimidine for a purine, then the mutation is called a transversion. If a pyrimidine base is substituted for another pyrimidine base or a purine for a purine, then the mutation is a transition. So what happens if Charmeleon's mRNA ends up having a single point mutation? Let's say that cytosine substitutes guanine in the 3rd amino acid, so a transversion takes place. This will result in a missense mutation, which means that the amino acid will change from arginine to serine. Well, with this mutation, Charmeleon's Pokemon type will result in water instead of fire. Another single point mutation that can happen is a nonsense mutation. Nonsense mutations change a normal codon to a stop codon, which shortens the length of the protein. Here, the first adenine in lysine is changed to a uracil, so it's considered a transversion. Since Charmeleon's protein was stopped short, so will its evolution, so it will actually be Charmander instead. The third single point mutation is a silent mutation. Silent mutations code for the same amino acid, so typically a drastic change to the organism will not occur. In this example, the cytosine in phenylalanine is changed to a uracil. This transition of bases still results in the phenylalanine amino acid, so Charmeleon will undergo a small mutation of being able to perform 3 more attacks than it normally would at its state of evolution. Lastly, a read-through mutation can occur. In read-through mutations, the stop codon is changed to a codon that codes for an amino acid, resulting in a longer protein. In this example, I swapped the adenine and the stop codon for a uracil, resulting in the tyrosine amino acid. As you can see, this transversion of bases adds more amino acids, so since the protein is longer, Charmeleon will continue to evolve to Charizard. And these are my references.